In this video, I will be continuing on with the cell structure topic. I will be going over microscopy in relation to cells. We use microscopes to study organelles within cells as they are too small to visualize with our eyes. In your exam, you need to know about 3 types of microscopes and what the advantages and disadvantages are between them. As you can see from this table here, it summarizes everything you need to know. Let's get some terminology out of the way first. Magnification is how much larger the image is compared to the original image, so how magnified the image is. Whereas resolution is how well a microscope is able to distinguish between 2 points that are close together. So, the first microscope that you need to know is the light, or also known as the optical microscope. This uses light to form an image. You can only see the images in 2D and has a low maximum magnification of 1,500 times. The maximum resolution is 200 nanometers. This means that the distance to distinguish between 2 points is 200 nanometers. So the images are not that detailed and you may not be able to see smaller organelles like ribosomes or lysosomes. The main advantage for using a light microscope is that you can look at living specimens. Scanning electron microscope uses electron beams to form an image. The magnification is a lot higher, 200,000 times, and resolution of up to 20 nanometers. The advantages with using this is that you can view specimens in 3D and you can stain them to produce colored images. The optical microscope also produces colored images as these are living specimens, whereas with the scanning electron microscope you need to stain these specimens in order to gain the colored image. The transmission electron microscope still uses electron beams, and the specimens must be put into a vacuum, so you definitely cannot view living specimens. You can only view 2D images, but 2 million times the magnification and 0.1 nanometer resolution. So with transmission electron microscope, you can see organelles in great detail as the distance between 2 points is much smaller, hence a higher resolution. Now, in your exam, if they give you a magnification calculation question, for that, remember this triangle. If you want to calculate the image size, then cover that bit and you are left with the magnification times the object size. If they ask you to calculate the magnification, then cover the magnification and you're left with the image size divided by the object size. Also, remember your conversions. So 1 millimeter to a micrometer, you need to multiply by 1,000, or the other way around, then divide by 1,000. In order to convert 1 micrometer into a nanometer, you need to multiply by 1,000, or the other way around, then divide by 1,000. Now, in order to study one specific organelle, they need to be separated from one another via a process known as cell fractionation. Cell fractionation is split into 3 stages: homogenation, filtration, and ultracentrifugation. Homogenation is to break up the cell and to release the organelles using a homogenizer. So first, cells are placed into a solution. This solution has to be cold to reduce enzyme activity. It must be isotonic to ensure that water does not move into the cell and into the organelles via osmosis, which may cause them to expand and get damaged. Then it needs to be a buffered solution to ensure that the proteins within the organelles do not denature. So the solution containing the cells is then put into a homogenizer, which is like a blender, and this machine will grind the cells to break them up and break the plasma membrane to release the organelles into the solution. This new solution with the organelles is called the homogenate. Now the homogenate is filtered, so the large debris that wasn't broken up properly are removed. The remaining solution containing the organelles will go through ultracentrifugation, which is where the tube is placed in a centrifuge. This machine will spin the tube and separate out the organelles. They are filtered out in order of mass, from heaviest, for example the nuclei, to the lightest, for example ribosomes. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.